Great, thanks Shu for the introduction, and thanks to the organisers, especially Shu and Derek, for coming up with this format several years ago. And I remember, Shu, we were in Queensland, I think, and you said something like, it will be your turn next to Victoria. So we're very pleased to be hosting MND Connect this year. So today I'm going to be talking about stem cells, and I think most of you, when you think of stem cells, you probably think of a stem cell therapy or a stem cell transplant. But I won't be talking about that today. I'll be talking about using stem cells as a tool to search for new drugs for motor neuron disease. And what's useful about stem cells is we can actually convert them or turn them into motor neurons. And what you can see in that slide, those green cells, are human spinal motor neurons that we've grown in the laboratory. So you've already seen this slide today from our other speakers, but just to reiterate, the theme of today is MND, one disease, many faces. So we do certainly appreciate, as clinicians and also researchers, that MND comes in many forms. There are many presentations, each case is unique, and I've illustrated a couple of forms here. So here we've got spinal onset, we've got bulbar onset, we have the other forms of PMA and PLS that we've heard about, and also different patterns of presentation. So we have symmetrical weakness, asymmetrical, we have our variants of PMA, the flail arm and the flail leg. So this does present a challenge in terms of designing effective treatments for this disease and also obviously managing the disease. And again, this is that spaghetti plot we saw before earlier today. This is the clinical course of these patients. So here we're looking at a clinical rating scale over time, and these traces are individual patients with motor neuron disease, and you can see there are different trajectories in these curves. So we have patients who succumb to the disease in the classic 1 to 2 year period. There's also a patient that managed to get out to 7 years, and you can see that there are different rates of progression. So that this makes the disease quite challenging from a research perspective to model. And I would say that over the last 15 years of my research in this area, we've really focused almost exclusively on the genetic form of motor neuron disease, which is the familial MND. So if you look at this pie chart here, we've been focusing on the genetic form, and there hasn't really been done much in this space, which is the sporadic MND, which Ken referred to before. So that's the form that's out of the blue. And I would say in the last 25 years, most of the research community has focused actually on this slice of the pie, which is 2% of all of MND, which is accounted for by a mutation in this gene called SOD1. And subsequently, a few years ago, there were some questions raised, at least historically, about how this mouse model was used. So this is a genetically engineered mouse that carries this human SOD1 gene. And what the realization is, a lot of the effects, a lot of the positive effects, positive drug outcomes in these animals were lost in translation. They simply did not translate to people with motor neuron disease. And the reasons for this are obvious. So there are clear species differences between mice and humans, but also this mouse is replicating only 2% of all of motor neuron disease. It doesn't really mirror the sporadic motor neuron disease. So we need to come up with radical new approaches as to how we can tap into sporadic motor neuron disease and also account for that disease heterogeneity or variability. And I just wanted to show this slide, and Dr. Maith has touched on this before, and I think one of the issues we have in the way we approach clinical trials is we assume this one-size-fits-all model. So if we come back to our patient population here, we have a mixed population of different forms of motor neuron disease. And what we will do is we will take one drug, so in this case, drug A, we will treat these patients, and there are usually 3 outcomes from this. So in some cases, you'll have an adverse event. So these patients will drop out of the trial, they cannot tolerate the drug. In the majority of patients, you see 0 benefit. There's no clear clinical benefit. But perhaps in a subset of patients, there is a benefit. So this is the green subgroup here. But the problem in the way we conduct clinical trials currently is these patients are all combined, and you end up with these similar plots. So the real drug effect gets lost in the noise or in this noisy background of motor neuron disease. And what I wanted to move on to now is we need to come up with a new approach, and there's an innovative approach taking shape, and it's been happening for a while in cancer and hematology. And it's this concept of using individualised treatment plans or personalised medicine. So how does this work? Well, we start off with this mixed population of MND patients, and what we can do is we begin to profile them. And what I mean by profile is it's almost like you start doing genetic testing and you start to barcode them or split them off into various subgroups of motor neuron disease. And then by doing that, you can actually start to tailor treatments to those specific subtypes of motor neuron disease. So in this case, we have treatments A, B, and C. And by doing this, by stratifying our patients into these groups and giving them individualised, personalised treatments, you have a scenario in which now every person is able to benefit from their treatment. So how can we go about this as researchers? So we were very pleased last year to launch a program, an exciting new program called the Fight MND Drug Screening Program. It was funded clearly by the Fight MND Foundation. And this is just a slide just to take you through how the program works. And there may be some of you in the audience who have participated in this program. And what we've done is we've taken 150 MND patients. They've been anonymised, so they've been coded. We've collected them from, this is a interstate program, mainly from Victoria, but we did open it up to other states. And these patients gave us samples, very precious samples. In this case, they were skin samples. And these samples were then turned into stem cells. And we outsourced this to the Eye and Ear Hospital in Melbourne because they've been doing this for years on blindness conditions and also dementia. And they're able to provide us with these stem cells from these MND patients. They're given to the Florey. We do a bit of QC with some checks on them. We're able to convert them into motor neurons and we start to profile them and eventually get down to business, which is this drug screening. And the ultimate goal is we can pull out a hit and then bring this back to the patients through various funding agencies such as Neuroscience Trials Australia and Fight MND. So how are we doing this? What's the technology driving this? Well, what we're doing is a very well-established technique in which the patients or the donors in the program, they're able to provide a superficial skin biopsy from the forearm in which we're able to grow these skin cells in culture. We're able to use some molecular tricks to turn them back in time to stem cells. So these are now the progenitor cells of life. And then we're able to coax them into becoming brain cells. So in this case, we're interested in motor neurons. And I wanted to just show you some examples of these cells from the laboratory. So here we have some skin cells that we've taken from MND patients and we've been growing them here for about 2 weeks. So you can see that they grow like weeds and they don't really look that quite interesting until we turn them into motor neurons. And then in a process which takes about 3 months, we're able to turn those skin cells into stem cells. And again, this is an example of how the stem cells appear under the microscope. So they form these large colonies of cells. And then we're able to add chemicals and use some molecular tricks and turn them into motor neurons. So here you're looking at some examples of very beautiful, pretty looking spinal motor neurons grown from MND patients in the dish. So here, this is a period that takes about 1 month, so we're now out at 4 months. What are we doing with these cells? So now we've got access to motor neurons from patients living with motor neuron disease, so this is quite unprecedented technology. We can now generate infinite numbers of these cells. What are we doing with them? So what we've done is we've built an amazing platform, and as many of you know, there is a lab tour at the conclusion of MND Connect. You're able to see this machinery and this pipeline in action today, so I do encourage you to attend. Everyone is welcome. We've got some registrants, but it is open for everyone here. And what you'll see examples of today is we're able to take these motor neurons that we've grown from MND patients, we're able to add them to these plastic dishes, and you can see that these dishes contain tiny little chambers, and there's about 384 of those per plate. We're able to then scale this up and put many plates into these machines. And this is the robot. This is the first robot you may see today. We call this a drug treatment robot. So this was fully customised and built for this specific purpose for motor neuron disease. And we're taking drug libraries. So drug libraries are commercially available, drug libraries available from drug companies, and these contain thousands of compounds. Our primary focus is on prescription medication and clinical trial drugs. because we believe that this is a form of drug repurposing, and this will actually accelerate the transition to clinical trials. So we're taking these drug libraries, and what we're doing is we're using this drug treatment robot to treat these cells in order to improve their health and survival. And then we move over to this second robot, which you may see today, and we call this the drug screening robot. And this is actually a very fancy, high-powered microscope. It's able to image these cells live and document all of these different features of cells that we're interested in. And the hope of this program, the goal is to pull out at least one drug hit from these chemical libraries can treat motor neuron disease effectively in the dish. And as I mentioned, we call this drug repurposing. The rationale behind this is because if you take a prescription medication that's already available in the public domain or the medicine cabinet, you can then take it straight into a phase 2 efficacy trial. You don't need to worry about safety. So I've just got a video here that I'll play. This is a time-lapse video of the drug treatment robot in action. So what this robot's able to do is retrieve the cells in that plastic dish on the left of the screen, and then it's able to essentially squirt on the different chemicals or the drug libraries onto these cells. So this is a very clever operating system. This is not possible to do this manually, and we can even get the machine to spell out words such as our donor behind the program. So this is great promotional material. So this is large scale, it's accelerated, it's fast paced, and I would even say this is industrial scale. And this is exactly the pace that we need to tackle this disease. Now this is an example of a video. Here we're looking at some motor neurons. We've coloured them with a glowing green dye, and we're able to watch these cells over time. It may not be very clear in this video, but you'll get some good examples today on the tour. We're able to see these cells behave. So these motor neurons are quite mobile. They form these long connections. These connections get disrupted in MND, and we're looking for drugs to really support and maintain those cells. So this is another way we can look at it. This is a cartoon. What we're trying to do here is establish motor neuron disease in the dish, and we know that these cells begin healthy, and over time they start to lose their healthy glow and fluorescence, and they take on the sickness of the disease. And essentially, this is almost a binary system. We're looking for cells that can survive versus cells that lose that glow over time. And we're essentially constructing these types of curves. So these are, we saw examples of these earlier today. This is a survival plot of these cells over time. And time in this system is measured by days. And we can see that these control cells from a control healthy individual begin to die over time. And we know that the motor neuron disease affected cells have a different survival. And in fact, what you can see here is the survival curve is shifts towards the left. The aim of this program is to intervene and to stabilise the health and survival of these cells. So put them back into this direction. And again, this is another curve showing here's our untreated motor neuron disease cells, and we've applied a drug here. We're able to shift that survival curve towards the right. So now we're supporting the life of these cells. And what's great and exciting about this program is it really gives us the opportunity to scale up the number of drugs that we can test, because we know that this disease will require perhaps a cocktail of factors and treatment approaches to defeat. So here we're looking at the additive effects of different drugs. And what we're doing is really analogous to classic clinical trials. This is the one where riluzole was tested about 20 odd years ago. You can see that riluzole was effective versus the placebo group. This is exactly the process that we're emulating, and I would even call this a clinical trial in the dish. Now, how are we tracking in time? So here's a local who dropped in, this Neil Danaher, as you know, is one of the founders of Fight MND, and he was our first donor, patient 0 in the program. So here he is getting his skin biopsy, and his cells are cultured in our laboratory. Here we've got Chris, so you'll meet Chris today. Chris will give you a demonstration of the robots if you go on the tour. And here we've got some of the Justice League from Bethlehem, so we've got Emma and Ruth. And it's been a really great collaborative project with various hospitals, including Bethlehem. Here we've got Chris checking out the machine, the drug treatment robot. This is Catherine, so you'll see Catherine today if you're on the tour. So we've got a very dedicated team working around the clock on this program. And here are Chris and Catherine analysing some results from the second robot. So I just wanted to tell you briefly today, how are we tracking in time? Our goal was to recruit 150 MND patients, and we achieved that back in July and August. So we've hit our goal, and I just wanted to say thanks to MND Victoria and MND Australia for really helping us with that recruitment phase. And we're really now in this phase where we've received 100 of those stem cells from those patients, and we're doing very rigorous quality control and checks before we begin our drug screening. As I mentioned, it's a national program. It's Victorian-centric. We did open it up interstate. We've even had patients flying from Perth and Hobart at their own cost to be part of the program, which was wonderful. And just a couple of pie charts to show our emphasis in this program was sporadic motor neuron disease. We really want to focus on that part of the pie chart, and we've certainly recruited both the familial genetic forms of MND patients and also sporadic motor neuron disease. What have we recruited in terms of sex? We've got mainly male subjects, as you would expect, that this disease is 50% more common in males, so we can look at sex differences. This is a pie chart showing you age of onset. So while this disease is certainly more common in middle life, we have recruited that whole spectrum of age of onset, which was the goal that we set out to do. And again, diagnosis. So we've captured mainly ALS patients, but we do have these other forms of MND, PLS, PMA, and PPB. So again, this comes back to we want to study the whole disease phenotype and heterogeneity. And then lastly, this slide just shows you we've got patients with all forms and all presentations of MND. So we've got the limb onset, the bulbar, respiratory, generalised. We've got slow, classic and fast progressors. And these are the genetic forms of MND that we've captured. So we've got some of the classical SOD1 C9 genes. So I just wanted to end by saying we're very excited to deliver this program. Clearly, the short-term goal is to screen these libraries and find prescription medications that we can perhaps repurpose for MND. But this program really does open us up to other avenues, and that is one can start thinking about performing true precision medicine. So true precision medicine requires you to know that the whole genetic and molecular makeup of these patients in order to predict why they respond positively to a drug. But we're even getting more fancy. There's a system now where you can create these so-called mini brains in the dish, so you can go 3D and also interface them with chip technology. So these are really exciting directions for the program. I just wanted to close by thanking a huge cast of members, especially Dr. Mathers and her team at Bethlehem and Paul Torman. Everyone's jumped aboard, obviously Fight MND, who are the major sponsors of this program. And I just wanted to end with this slide. I really wanted to dedicate this program to Dr. Ian Davis, who was a friend to many of us all, and it was actually Ian's vision and he really drove us to get the funding for the program. And Ian's message to me was always think big, think national, think global, and that's exactly the direction we want to take this project. So thank you.